presentation in the FishCAST seminar series. My name is Dr. Christina Semeniuk. I'm from the University of Windsor, and I am the director of FishCAST. And I am absolutely delighted today to present to you and to introduce to you our speaker, Dr. Catherine Febria. So Catherine Febria, she's an assistant professor here at the University of Windsor. She's cross-appointed between the Great Lakes Institute for Environmental Research, as well as the Department of Integrative Biology. Dr. Febria also holds a Tier 2 Canada Research Chair in Freshwater Restoration Ecology. And so how did Catherine come to acquire this illustrious career? Well, she did and started off first with her bachelor's in science and environmental science at the University of Toronto. She then moved across the country and she did her master's in geography at Simon Fraser University, where we actually overlapped. We were in different departments, but right, just like ships passing in the night, Catherine. Afterwards, Catherine jumped back to the University of Toronto, where she obtained her PhD in ecology and evolutionary biology. She also obtained a collaborative certificate in environmental studies as well. Upon the completion of Dr. Febria's doctoral degree, she became a research assistant at Chesapeake Biological Laboratory at the University of Maryland. And then she got the draw and the pull to head to the Southern Hemisphere, where she was at first a postdoctoral fellow, but then became the director and the main scientist of the Canterbury Waterway Rehabilitation Experiment, called CAREX, the acronym, and that was part of the Freshwater Ecology Research Group at the University of Canterbury. Well, the University of Windsor put out a call for someone, a really high-end ecologist, a restoration ecologist, which was attached to a Tier 2 Canada Research Chair. And when Catherine applied, it definitely was a no-brainer, and we are so happy to have her here. And when Catherine started at university, this is when she founded the Healthy Headwaters Lab. So she's a founding member where her and her lab address challenges that are facing freshwater issues around the world. And her lab addressed these issues by drawing on robust data synthesis, empirical place-based studies, as well, and really importantly, translational ecology. Her and her lab's close work with communities and decision makers are what helps to drive her truly impactful science. And if there is something that you should know about Dr. Febria and her lab and the members and who she works with, collectively, they're all committed to inclusion, diversity, kindness, and curiosity, and it makes up their mandate and their mantra, and it's part of their commitment to science excellence and impact. And of course, I am so pleased to have Dr. Catherine Febria be the Associate Director as well of FishCAST. And so without further ado, I'd like to introduce you to Dr. Febria, where she'll be telling us today about centering communities in freshwater restoration science. And with that, Catherine, I'm going to head and turn it over to you. So thank you so much for presenting for us today.
Thank you, Tina. Hopefully you can hear me. And audio's good, great. Thank you so much for that introduction. It's really humbling just to have, hold space with Tina and the FishCAST group of PIs, partners, and trainees. It's been a wonderful experience so far, and I really wanted to just extend my gratitude to everyone that chose to spend their afternoon or their morning or evening or wherever you are in the world today with us. And hopefully you'll have some insights into the research that I do, but also really wanting to speak to partners, trainees, potential or existing trainees in the program to understand the lens through which I bring and contribute to the training of our next generation of fisheries scientists, managers, and partners. Let me make sure I can. Okay, so I've just moved to my next screen, and I first just wanted to start by addressing my own positionality. Today I'm standing here at Waiwayatong Zibi, or otherwise known as Windsor, the Southwest Ontario pocket here in the Laurentian Great Lakes, here in Turtle Island in North America. My story and my relationship with water certainly didn't begin here and is placed here now, but it was certainly through this relationship to water and its stewardship that I'm here with you today and that I do the work that we do. I'm really privileged and grateful to be an immigrant settler from the colonized islands of the Philippines. And the work that I'll be talking to you about today will take you not only to Aotearoa, New Zealand, where I had the privilege of working for the last 5 years previous to this position, as well as to the Chesapeake Bay in the North America, and here in North America. And I'll be hopefully, if you will indulge me, I'll be able to talk to you and give you examples of how centering communities and the knowledges that these communities bring in freshwater restoration is absolutely essential to accelerating and advancing both the science that we need and to accelerate the practice that needs to happen. And I really do, just based on all of the evidence and lived experience, believe that early career training is essential to achieving this vision of future planetary sustainability. So hopefully you'll get some ideas on how we can rethink and reframe partnerships in fisheries science, research, and management, really starting from the bottom up. And in so many different ways, you'll see just flavors of that throughout today's talk. I really aim to be, I practiced it, tried it to make it a 30-minute talk, but it will go over. So if you get nothing out of, or if you have to leave early, it will be recorded. And I hope that you sort of get the gist of this through the examples that I'll provide today. So as Tina mentioned, the lab is quite a new one. We were established in 2019. We called ourselves the Healthy Headwaters Lab because we wanted to be very mission and values driven. Our goal is to use science to connect land and water and people for the benefit of future generations. And so by starting out with this vision, you can see that we have a key values that drive our work. We are very intentionally anti or decolonial in the way that the science is being developed and advanced. And we are intentionally interdisciplinary because that is the challenge of the science and the solutions that we're asked to address. There is a community centering in this work, and I'll explain to you in so many different reasons why this is the essential way to proceed if we want to achieve restoration. This all is under an umbrella of, language and terms that we're hearing around transformative change. How can we lever the kinds of knowledges and expertise in different corners of where we are living to really accelerate and promote change and to achieve our goals for sustainability? This will all come together by trying to achieve beneficial societal outcomes. And all of these threads underpin what we know as excellence in research. But I'm here today not as a fisheries person. I've always loved small streams and wetlands. This image from Judy Meyer's paper in 1997 in JARA really helps remind us all about the importance of these smallest streams, wetlands, and intermittent waterways that are often bulldozed and buried for the purposes of human activity. We know that these small waterways are the hotspots of biodiversity, whether they be benthic invertebrates or hotspots of nutrient cycling, such as the export of organic matter and nutrient transformations that support fisheries downstream. And likewise, we know that these river networks are connect intimately with the different fish communities that come up to the headwaters for critical parts of their life history. And yet, despite how the numerous studies and research that we have on these wonderful waterway networks and river networks, we still continue to talk about and have to validate why we need to care about them. And that, goes back for many years now about trying to generate some place-based data on these intermittent streams and making them count in the sense of in global climate change models, or in protection from things like suburban development or land development or agricultural land use development. This is one of these things that come up in the news of reasons why we need to protect them. And the evidence is really, there's so much evidence out there that it really ends up becoming a really socio-political conversation and not a scientific one. But the idea that ecosystems and these headwater streams is being ultimately connected to these larger waterways and these and aquatic and terrestrial ecosystems goes way back to its the first definition of ecosystem. Arthur Tansley in his 1935 essay identified this idea or defined ecosystems as being the ones that are the most various of sizes, from the universe as a whole down to the whole atom, to the single atom. And that these systems we isolate mentally are included as parts of larger ones, and they overlap and interlock and interact with one another. And that is what kind of, it really captured my imagination as a scientist to understand how do they overlap, how do they interlock and interact with one another. And, in 1935, there was an acknowledgement then that the isolation was artificial, but the only way to proceed. And I'd like to argue that today, almost 100 years later, we now have enough knowledges, and it was really understanding how things connect as the real challenge for headwater stream ecologists. And so when we think about the Great Lakes, many of you may think about the fisheries, but I think about the trees, I think about the wetlands, and I think about the people and the relationships that we have around our relationship to water and land, and in restoring our relationships to these places. And this was this photo here is of me and my eldest a decade ago in an old quarry in the city of Toronto, which is now an active, vibrant place for nature, for community building, and for ecosystem functions that, you know, really trying to advance science that embraces all of those dimensions as being the necessary way of protecting these small waterways, these critical fish habitats. So let me first begin by talking about what we know about freshwater restoration science. And again, I'm focusing on these small streams and river and stream ecosystems because they are the critical fish habitats. I often work in partnership with fisheries scientists and managers and restoration and geomorphologists, experts, to really understand how can we restore those functions and biodiversities that these systems support. And, you know, far and wide, there are many citations of work that have recognized that, like, for example, Emily Bernhardt's paper from 2005 that synthesized all of these restoration projects around the US to show that it's a very, very lucrative industry to invest in restoration, but there's very, very little evidence of success. And when we actually looked at restoration projects, more than 500 that took place in North America, from freshwater and coastal wetlands and waterways, there was no consensus on what kinds of things we should be measuring and what combination of metrics best equate the restoration and the return of the desired ecosystem functions and biodiversity. And there are a lot of reasons for this, and really trying to understand why it comes back down to what are the types of activities that are being taking place. So if we think about an agricultural landscape, one of the most common human impacted landscapes on the planet, most of the activities often end up being something like riparian planting. They might also involve something like at the edge of the field. And maybe even moving into the channel margins to create maybe an artificial floodplain. And if we're lucky, in some of the more developed countries, there's been a long history of reconstructing whole streams to have meanders and features that would promote things like organic matter buildup, oxygenated places for fish to occupy in their habitats. And so this, regardless of this wide range of tools that have been applied around the world for almost a century now, ecosystems continue to be degraded, even after decades or generation or two of reforestation and replanting. Some call this saying that we're in recovery debt. And there's also a highlight that, you know, with as much science and activity as we have across these different ecosystems, we really need to accelerate the kinds of actions, both the science and the practice of restoration. So one way that our lab and my research has really tried to address this is really start to think about the function of restorations. What kinds of functional indicators can we use to help guide and accelerate restoration? We can almost envision that these drainage systems, these waterways that are often channelized as drains in cities and agricultural landscapes, the biodiversity and ecosystem functions are literally stuck in a ditch. There's this concept or this theory that we're testing around healthy versus unhealthy resilience in degraded ecosystems. And we can go back to some of that theory when we think about what is the goal of restoration. Sam Lake says it in this really nice paper around how the idea of restoration is to engender both the positive resistance and resilience attributes in a restored system, overcome through our intervention and restoration activities the negative resistance that really pulls our ecosystems into this degraded state and keeps it locked there. And so using things like restoration efforts that have happened on the ground now, but also data sets from across a broader gradient of space and time, we can really start to think about not only what species are there, but the functional traits that they offer. And what we are aiming to do is pull our systems, and looking here at this food web from a Canterbury waterway, which is very similar in nature in terms of being populated by dominant generalist taxa like invertebrate worms, chironomids, and snails, to shift through restoration, continued and intentional restoration activities to promote higher trophic levels like fish of cultural and societal value. So we can go ahead and test this, which is what we've done in the last couple of years in Aotearoa, New Zealand, creating stream channels up in the highlands of the Southern Alps, really trying to confer traits between different trophic levels and nutrient transfers across them to try and test this. And we can certainly confer that. And so the question then is, once we're able to figure out what are those mechanisms of biological delay, what kinds of tools can we use to overcome that resistance? And so this is kind of the theme of a lot of the scientific projects that we do. But if we really want to think about planetary sustainability and really achieving freshwater restoration, we again can look to the published literature. And when we look at examples of those restoration projects that have happened around the world, the ones that have the best chance of achieving long-term sustainability are ones that include and embrace the societal dimensions of restoration. So there was a lot of this, and I've been privileged to engage in a lot of community engagement, working with decision makers and individual farmers and local knowledge holders and rights holders to really understand the context within which restoration is being placed. And this echoes what we're hearing from around the world and also here in Canada. When we can incorporate local and Indigenous perspectives, we are guaranteed or more likely to achieve impactful research and effective management outcomes. Some of the best case examples are right here in Canada with our First Nation partners. So this sort of recognition is also coming at a time in society and in the world where there are increasing calls for coordinated action. I'm sure, like you and me, during this pandemic, we had seen so many different reports around the status of our world's ecosystems. At the same time, our relationship to nature was probably what kept us really sane throughout this whole pandemic and this ongoing pandemic. And so at that same time, various reports from the UN and IPCC came out reminding us that the world is getting hungrier, the land is getting exhausted, and we need coordinated action to curb soil erosion. So again, I'm a freshwater ecologist and I'm looking at this, and where does that soil go? Well, they're going into our rivers and streams and wetlands and into our oceans and coastal systems. At the same time, another group that we work with, IPBES, the UN Platform on Biodiversity and Ecosystem Services, released a similar echoing complementary report that humans are responsible for driving more than 1 million species to extinction. So the question now is, what are we prepared to do? And we need to really rethink and reframe the ways in which we are pursuing the science and practice of restoration. So maybe some of you, like me, have been attending the Society of Ecological Restoration's global conference this week, and it's officially the launch of this UN Decade of Ecosystem Restoration. So for the next decade, there is a global focus and intention to really accelerate both the science and practice of the efforts and coordinate actions on the ground. And so I'm hoping that through the next couple of examples, you'll see ways in which we can do this, and it looks different in every single research partnership. And so my first example really comes from, more than a decade ago when I really made the intention to really think about how can we make our science actionable. I was privileged enough to spend time at the launch of the NSF, the US National Socio-Environmental Synthesis Center, as part of my postdoc, where we had intentional conversations about how can you make science actionable. And so what is actionable science? Dr. Semeniuk talked about it as translation ecology, which is a term we often use. It's also it really is an intentional process where we are trying to identify a knowledge gap. What is the science question about this particular environmental decision-making context? And by including these diverse knowledge systems as part of that process, and not like the stakeholder at the end, but really as part of our science team, we can really clearly identify the uncertainty and really help guide which type of science is needed for that type of problem. It's a very decentered knowledge production process. It's a very non-hierarchical process that allows us to co-innovate solutions to really answer the question that needs to be asked, and not the science question that my skills might be better suited for. So there is a nice summary of this by Nordstrom and a number of colleagues globally that identify that all of these terms fall under all of the ways in which we do this translation ecology, actionable science. It's this umbrella term of knowledge co-production and sustainability research. And essentially these 4 boxes capture the sorts of elements that take place in every single project that we undertake. Really thinking about, sure, here's the solution. What is the local context? What are the challenges? Who are the rights holders involved? Who are the communities involved? And have we engaged all of those knowledge systems to really help define and drive our research? And that is a real iterative process. Can we identify a goal that's one that our team might address, but maybe is a question for another science team to address? But having a process in place, an intentional research methodology like co-production, ensures that we get there. And that if things should fail or if things should change, because we've engaged in this process, there's no failure, but really an opportunity to figure out what is the next step. So speaking just about science and a science kind of partnership example, in the Chesapeake Bay watershed case, I just want to talk a little bit about what we were able to do with other types of scientists. Funded by the US EPA and NOAA during the time of Barack Obama's administration, there was a real interest in understanding how well stream and stormwater management practices in the Chesapeake Bay area, which provides a significant amount of water purification and fishery value and services for the Chesapeake Bay, how well do stormwater management tools work in the face of climate change, recognizing that there are more nutrients being loaded into the Chesapeake Bay and that there are different regulatory tools in place. So we engaged with all of the data that we could find for watersheds in this area. NOAA and EPA scientists that had been monitoring stormwater ponds and different common best management practices in the area. And then we also decided to engage with mathematicians or decision scientists to help quantify the uncertainty, as well as climate scientists to help generate climate change predictions so that we could actually model and export nitrogen loads out of the BMPs into the bay under future climate change scenarios. It started with gathering the data through synthesis, looking at how many papers we were pointed towards. And it was as from an ecologist lens, we were able to identify that out of 10,000 entries in this incredible international database, only 30 of them offered data that were relevant in terms of ecological factors that drive nutrient retention versus release under a stormwater event. We were able to then engage stormwater experts from around the bay who we could give data and use their own knowledges to generate distributions of data for us that we could then go ahead and model. And so you can read about this in that paper. It's really just to highlight that we can generate new methodologies or approaches by engaging different types of scientists to help not only include uncertainty in the process, but quantify it so that at least a decision maker can go ahead and model it or use it in their decision-making frameworks. So one example was that we were very, very often in BMP spaces, legislators and regulators, they'll often want to know what is the BMP's efficiency? And so if you look at this box and whisker plot, you can see here in nitrate, usually in a lot of regulation, they'll use 30% reduction of nitrate in a wet pond. That's the number that they'll attribute in terms of reduction. But when we actually took the numbers and calculated the width of the variability around it, you can see that it goes above plus or minus 100%. And so, we could talk about, the questions could say, well, it's 30%, and we could be surprised as to why there's more nutrients being loaded into our watersheds, off our watersheds into the main coastal waterways, or we can actually plan and include that variability in our estimates and really start thinking about retrofitting and different types of management options in the face of climate change. So in another example, I wanted to now switch and talk about community-led and community-driven restoration. So increasingly, we're starting to hear things like networked and embedded and aligned experimentation. And in my lived experience and of the data that we've been able to generate, it's really been clear that this is our a really important way to really accelerate experiments in the real world. We can do so much, we can only do so much in our universities or in our experimental chambers and that kind of thing, but really thinking about moving our experiments into the real world is something I spent the better part of the decade doing. It is an approach that also recognizes that, again, if we look at the literature, restoration takes time. It takes decades, a generation or more to really restore these processes, our communities, and the functions and biodiversity. And so investing in the social infrastructure needed to nurture these ecosystems back is an obvious, incredibly important part of our research process, and really an important part of ensuring that the science continues to be monitored and promoted beyond, a 2 or 3 or 5 year, if you're lucky, research grant. So to illustrate this, I'll take you to Aotearoa, New Zealand, and in particular Te Waiponamu, the South Island, and Waitaha, the Canterbury, the lowland plains of Canterbury. For those of you that think about New Zealand, you may think about these incredibly beautiful, endangered, braided floodplain systems, one of the few in the world, and hobbits and really happy cows, and that certainly is part of that experience. But also we have a landscape that's very similar to the Great Lakes and where I am here in the Lake Erie watersheds, where because of agricultural intensification, this otherwise native swampland has been channelized and converted to support intense agricultural practices. By channelizing wetlands, by burying wetlands and channelizing river networks to support a main economy for the country. It's also generated pollution issues, and in particular high nutrients and loss of very important culturally and recreational waterways and species. And so we took up the challenge to develop a restoration experiment, a program over 5 years that was placed and fully executed on individual farm waterways. And this is myself and a graduate student here monitoring some of these agricultural drains. We called it CAREX as a really just a cheeky acronym to remind people what type of plant, a CAREX secta, a sedge, that we were using to test an ecological theory around competition. And we engaged more than just numerous stakeholders. We also, and the farming partners, we engaged Indigenous rights holder Mana Whenua for the region, industry, government, as anyone that wanted to engage in finding a solution and have evidence to support it was welcomed, as well as the public. And I'll explain to you a little bit how it was intentionally laid out. And so we spent a lot of time talking to farmers, almost 18 months, myself and a restoration practitioner, just understanding the local context to devise a toolbox that, at each point of intervention, we could test using various projects and graduate student theses to really disentangle. And so we moved at tools like the bioreactor, which is pioneered here in Southwestern Ontario for the first time in Canterbury, riparian planting, again, testing some ecological theory concepts, channel margins to think about two-stage channels, and biodiversity additions in the streams. So this waterway could be anywhere in the Great Lakes Basin. It happens to be in Canterbury. And so I've shown this in a number of different talks before. Well, the pictures speak for themselves, where we were able to reveal the drain and waterway and measure biodiversity, really think about recreating a more natural channel margin, engaging the public to come out and plant the species, and over a number of different trials, you can see some experimental tries throughout the reach, carry out research on this space to identify practices and have a quantification of the different effectiveness associated with more than 7 tools used to manage waterways for invasive weeds. And so that's Katie. Over 3 years, she was able to demonstrate that the natural solution, riparian plants for which farmers could apply for grants to support, worked effectively at keeping these weed issues at bay while also providing biodiversity benefits. These trees on the sunny side of the drain started self-seeding within 2 years, and it really is a story that continues to speak for itself. But this is an example of training early careers at this critical nexus. It's a really privilege to say that Katie had identified that wanting to work at this nexus was a goal of hers when she started her PhD. She now is a science lead for Living Water, one of our partners on that program, but a key organization that has scaled the findings of our research to 5 national catchments across Aotearoa. Funded by Fonterra, the largest economic industry in New Zealand, as well as the Federal Department of Conservation, Katie is working at that nexus to ensure that science is at the table when management decisions are being made. As well as Brandon, a researcher who came from Pennsylvania to join our team. Not only was interested in trialing the first ever bioreactor in Canterbury, New Zealand, but using it at these public drains as an opportunity to test the concept of how much carbon does it take if we deforested this whole landscape, how much carbon, if we can put it in onion bags in these drains, does it take to actually generate the kinds of nutrient reduction levels that we need or are expecting to meet these national bottom line standards? And the answer is quite a lot. It does help then ground conversations about how much do we think we can expect from restoration at the edges of these fields in terms of water quality and biodiversity recovery. And so we engaged in continual and ongoing knowledge mobilization and translation of the work. We did radio shows, we had a community storyteller, we had a restoration practitioner who together made part of our team. We generated newsletters. For every farmer that we worked with, we generated a private document of the results of the biomonitoring data that we were working on and that we shared with them. A lot of the papers never really need to identify their name, so their privacy was always intact, and we had individual memoranda of understanding with each individual farmer that we were working with. And so, it really was about building trust and keeping that trust throughout. And to this day, the toolbox handouts that we created continue to be downloaded and available, so anyone can go ahead and check them out. It was really one of these comments where we'd hear from like a regional council, like, I don't read a lot of papers, but your newsletters are the ones that I just get the quick tips that I need to know about what grant I need to apply for, or what does it look like, and attend the demonstration sites. And that was more than enough for them to understand how they could implement it in their own practice. But not losing sight of the science opportunities that happen when they're place-based. So when Katie's waterways that she was monitoring went dry, we were able to participate in a broader global analysis of intermittent streams and the types of carbon being emitted out of these waterways. And this is previously, adding to a previous gap in knowledge around the impact that intermittent streams have on our global carbon cycle. So on one hand, we're doing a lot of the restoration, but still being able to engage and contribute to the need for a better understanding of how these systems are structured and how they function. But probably the best story that I enjoy talking about is the fact that we were able to do this through leveraging partnerships across to overcome barriers of scale. And that continues to be the biggest challenge in restoration. One project that you do in one area may not strategically be the place that is best connected to maybe a colony population downstream, or the riparian planting is downstream when what we really needed is upstream. And so this was a great example of how, from all of these different partners, we were all able to co-contribute and leverage our work so that not only did this 900 meters of waterway that we were managing achieve the kinds of things we published about, but there was a wetland upstream that we were able to amplify and bolster and find funding and help co-apply funding with the farmers. Or that another nonprofit took up the aim of writing a grant on a tributary right next door and funded more than 4 kilometers of additional waterway. And that the school downstream did a crowdsourcing thing and they raised money to plant another section. And then we also used the opportunity to build community. So my colleague and I, Mananui Ramsden, and I were able to hold our farmer conversations where we talked about the cultural value of these wetlands, stories that were placed on and how sacred these places are. And this as part of an effort to show that if you're continuing to steward this landscape and choose practices that are very much thinking about, farming and environmental sustainability, then it is achieving a common goal, a shared goal around ensuring sustainability for future generations. So this was all happening at the same time as the science community was going through larger conversations, at least in Aotearoa, about how to respond to and be responsive to our responsibilities under the Te Reti o Waitangi, the founding document of Aotearoa, between Māori and non-Māori. And so across science communities in this picture here are 50% Māori scientists and non-Māori scientists from early career all the way up to senior career, where we wanted to really shift the conversation into what does it mean to do science that's impactful for communities and science. And we led and launched this Kindness in Science Collective, where it's an active living space where we're really talking to the different science organizations at all levels to think about how we can choose kindness and in this place achieve better science outcomes. This is, again, all along the backdrop of a lot of other things happening in the world. Recognition through growing data that research excellence is certainly not possible without the lens of justice, equity, diversity, and inclusion. And I echoed again globally through the UN IPBES program, where for the first time we were able to participate, these are my colleagues and I, in fellows at the table doing the science policy synthesis to ensure that knowledge is mobilized and overcoming barriers of things like privilege between the global north and south, between different countries, as well as across the different disciplines. And this continues to be a vibrant area of research and community building at the forefront of sustainability. So when we think about, again, restoration, there's a lot of the science bits there. I'm hoping that you can start to see why I keep landing on the place of a common thread in successful restoration is centering Indigenous and local knowledge. This is building on so many other narratives and examples and evidence in different ways of understanding this, that if we want to have impactful research and management, that centering and beginning with our local community is the best place to start. And in Canada, this is very loud and clear. And I think we've all heard the news about yet another residential school in the news, and I'm just still quite processing that news. But it's just another reminder that this is the context within which we live and work here in Canada. And there are numerous calls, both from the Truth and Reconciliation Commission of Canada, but also from the science community, that we need to do better. And one way is to integrate Indigenous knowledge in teaching and research by really thinking about what privileges we bring and how can we change our approaches to the way in which science is pursued. So I've chosen to really try and challenge the funding structures and reimagine how we build a science team and how to embed knowledge and reconciliation in everything that we do. So for this last bit, I just wanted to talk about here Turtle Island and the Great Lakes, which is the place that I call home. We think about freshwater mussels as one of our pilot projects, and really this understanding that they're kind of our favorite species. They just live in our waterways. They're doing their water filtration thing. Fish get all of the glory, but they're there, sequestering nutrients, filtering out pollutants, keeping these fish habitats stable. And there's a hotspot of biodiversity right here in the Great Lakes and the Sydenham River watershed. It's been a privilege to work with partners, FishCAST partners, the St. Clair Conservation Authority and Fisheries and Oceans Canada, to train our students to work alongside. And so for the first time in 15 years of surveying the Sydenham River watershed, we've brought those data together to talk about and explore and interrogate, what is the role of that freshwater mussel within this river tributary. What we are showing and finding is that species at risk are highly correlated with other taxa in the food web, the invertebrate taxa in particular. And we're starting to characterize the differences between the smaller tributaries and the larger rivers to help inform and advance restoration priorities in that watershed. It also means that we've invested in a local farm advisory group of both Indigenous and settler farmers to co-develop and advise our research. Again, remember that call to curb soil erosion into downstream freshwater ecosystems? Well, rather than us deciding what is the best scientific model or comparison or boxes to bin, we've engaged the local farmers to tell us more and help us interrogate this Brookston clay, this low gradient clay plain landscape that we're living and working on. This has allowed us to move beyond these binary associations of how we farm between conventional and no-till, but really broaden our gradient to demonstrate what is this realistic trajectory of restoration if we include non-agricultural spaces like a forest that once was agricultural landscape 80 years ago. If we use chemical characterization tools, so we're using a lot of carbon complexity tools in the lab to try and knit together a picture of restoration baseline for the region. And this also allows us to really start adding biodiversity into the conversation in places where we normally don't associate them, like the roadside drain, like the urban stormwater pond. And so again, through partnerships with the City of Windsor, St. Clair, IRCA, Nature Conservancy Canada, and others, we're able to mobilize and do this work. And so again, we come back to our Odeimin, our Indigenous Knowledge Circle, as the heart of all that we do. And so a lot of those holistic pictures that I talked to you and showed you earlier is very much like a framework that's used in so many other cultures, Indigenous cultures, around holism. And so we really think about that as a framework for how we teach, like when we do our field course at the Quechua Territory, when we build community and participate in ceremony and learning about corn and working alongside the community when we're invited to participate. And likewise, accept the invitation to train the next generation of ecological monitors and stewards, and embrace the different ways in which that knowledge is shared, including hosting an inaugural artist, Mariah Alexander, to really develop and help create graphics and information for us to talk about what is the traditional ecological knowledge for the Great Lakes Basin. So I wanted to end by talking about and inviting partners here today and trainees to really think about, have a really open mind about what it means to be a partner. Co-develop research that really thinks about the diverse ways that knowledge is formed and shared, and really think about the opportunity there is to examine data sets And thinking in this restoration or management context, how can we extend spatial and temporal gradients so that we don't have to monitor something for 80 years to know that it didn't work? How can we embrace science communication and knowledge mobilization translation from the start? So it's not something that's a piece of transaction that we do at the end, but it is embedded as part of our methodologies. And, as we begin and continue to, do the work of restoration, just recognize that you can't do it without investing in the relationships and the communities that also need restoration as well. So I hope by today's talk, you have some ideas that, when it comes to freshwater restoration science, we really don't have a lot of time to wait about thinking about the next best experiment, but really thinking about accelerating through rethinking and reframing how we do science. And so I'm so excited to be part of FishCAST, and I'm really humbled by the invitation from the broader group of PIs to really bring this vision to training our next generation of fisheries and aquatic science scientists, managers, and partners. So with that, I'd be happy to engage in any other questions you might have. I'm really looking forward to meeting our trainees a little bit more after this. And please follow our socials, because there is a little bit of some hypothesis testing happening on the back of it all. And we hope to tell you more stories about how we can reconnect our relationship with land, water, and people. Thank you.